Hi, my name is Aishi, and welcome to part 2 of our lecture on response in signal transduction. So let's say we have two different cell types, and they're exposed to the exact same conditions, yet they will react differently. They should be reacting differently because they're different cell types. They have different roles to play, and so they should be doing different things. Now, why is this? Well, the answer is they will react differently because they have different genes turned on. Each cell type will have a specific subset of genes that are expressed, and this will be different from cells of other cell types. Now, these different genes will cause different proteins to be produced, and these proteins include signal receptor proteins, relay proteins, response proteins, a lot of proteins that are involved in the signal transduction pathway. And what this means is that even though you might have the same ligand, see this little red ligand here that I've drawn in each of these 4 boxes, which represent different cell types, even though you have the same ligands in each of these 4 boxes, the end results will be different because the signal transduction pathway is different. In the first 3 here, we have the exact same signal receptor protein, this blue guy here, but in different cells, you might get different responses. You might get different relay proteins that are activated, and this will cause a difference in the end response. Now, in the third example, you can see that there is crosstalk between different pathways. You have one pathway here and one pathway here. And in fact, the signal from this pathway, from this green ligand that was originally drawn, is actually influencing how this relay molecule reacts. This might be activating or inhibiting this relay molecule here. And this is important because different types of cells will have different signal transduction pathways. And for example, one cell might have a signal receptor protein for a given ligand, while another cell doesn't have that signal receptor protein, so it doesn't even respond to that ligand. And that's how a lot of differences will occur. And in this fourth case here, you see a totally different signal receptor protein for the same ligand. So last time we briefly talked about the termination of the signal. Now, why is this important again? Well, you don't want your cell to be constantly responding to that signal from 10 years ago, right? Chances are the environment has changed now. There will be new signals. That same signal probably doesn't exist, or if it does exist, there have probably been times where it didn't exist. And so you want your cell to be able to respond to changes in the environment. How does the cell do this? Well, first off, there's the binding of the signaling molecules, the ligands, is reversible. So it's not like your ligand is bound to your receptor at all times. It will let go after a while. And also, the relay molecules will eventually return to their inactive forms. For example, if one molecule was active when it was phosphorylated, then other enzymes will come by and remove that phosphate group, and that relay molecule will become inactive again. Or if it was inactive when it was phosphorylated, well, a kinase might come by and phosphorylate that molecule, and it will become inactive. Now, G proteins also have an inherent GTPase activity, and they will hydrolyze GTP to GDP, and they will essentially inactivate themselves.